very much, David. It's a great pleasure to be here, as always. I love coming to KITP, as David said, and I didn't know he'd figured out why I was so helpful. This is going to be a very informal talk. There's no slides. I'm just nothing at all fancy. And you should let me encourage you to stop me at any time and ask questions. And you don't have to be polite. You could just yell out over here, and I'll stop and answer the questions. And in fact, I'm going to talk for a while, and then I'll stop and ask you if you have questions. So it'd be better if it's more of a dialogue than a talk that I give. Just before I came here, I Googled brain and computer, and Google gave me a little over 20 million answers, and it did this in a quarter of a second. Now that is what a computer can do, and it's absolutely amazing. And what computers are so good at is they are wonderful at solving these complicated problems and at going through massive amounts of data. They didn't give me every site on the web. They just gave me the 20 million that were relevant, and they had to sort through all the rest to find those 20 million. It took them a quarter of a second. It's absolutely amazing. And computers are starting to do things that we never thought they would do. They talk to us and they're getting pretty good at it. When you call to make an airline reservation, we talk to them. Many of us not me, but many people to dictate things. A lot of physicians dictate their notes with computers, and they get them right very well a lot of the time. And so every year they're doing things that seemed impossible in the past. In spite of this incredible power of computers, you all are carrying around a computer in your head that does things that no computer that's ever been made can possibly do. And I'm going to give you a quick example of that now. Probably many of you who have wanted to email a New York Times article to a friend or a relative, had to read some funny letters and type them in down below. And the letters look sort of like this. The reason, and this is what is called a CAPTCHA. This stands for Completely Automated Public Turing Machine to tell if, T is Turing Machine to tell if, computers and humans are different. The reason they do this is because they don't want some spammer with a computer to be sending emails to people. They don't want them to be sending articles to people. They don't want them to be signing up. And so what they do is they do something that is they give you a task to do that is pretty easy for you, and actually it's not always easy for me because I have a hard time reading, so I have to do it two or three times sometimes, but the computers can't do it at all. When you look around this room, you see a bunch of objects, a lot of people, a lot of faces, and you don't realize how incredibly hard that is. No computer in the world can look around this room and see the objects the way that you do. The very best ones can be programmed, can be taught to do specific things. They're getting pretty good at recognizing faces in a certain way now. But to be able just to see the objects in the room is a computationally incredibly hard problem that we do so easily and effortlessly that we don't even know it's a hard problem. But computers can't do it at all. This is a very simple kind of CAPTCHA. CAPTCHA is sort of a takeoff on gotcha. This is a very simple one, but there are lots of other ones that you can think of. So for example, you could have a picture that had 10 faces on it, and down below it says, click on Santa Claus's nose. Okay? All of us could do that with no effort at all, easier than this actually, and that's something that would be really hard for a computer to do. Or you could have a picture like this, and down below it would say, drag the weight in the middle, so that the seesaw tips the other way. That's something that you could teach a computer to do that if you knew what you were doing, but to give a whole bunch of things like this, the computer wouldn't have a chance. So what I'm going to try to do today is tell you some things that are different and the same about the way that computers and brains work. We know in some cases pretty much what actual mathematical computations parts of the brain are doing, and those are the same mathematical computations that computers do. So our visual cortex, the part of our brain that processes visual information, uses some of the same techniques that are used to compress television images. And so a lot of the things we do are what they do, but we do it in a very different way, and that's what I'm going to try to describe to you. And what I'm going to have to start doing is I have to start by teaching you some things about the brain so that I can have something to compare to what a computer does. If you look at your brain from the side, it looks sort of like this. This is the front and this is the back. And one of the things about the brain that's very important is that the functions that the brain performs are clustered together. This is the cortex of the brain, the surface on the outside, and I'm going to tell you after a while that it has about 10 billion nerve cells in it. And this has been divided up into about 100 distinct areas. And for example, there's an area back here which is for vision. There is an area here for movement. There's an area here for hearing. There's an area here and another area there for language. And you'll also hear later that if you kill brain cells at random, then the brain still functions quite well. But if you kill all of the brain cells in a particular area, like here, you can't see anymore, because all of everything about your brain that has to do with vision starts by that one part of the brain right there, the visual cortex, and then it sends information to about 24 other areas. So there are about 24 areas that are involved in vision. Yes.
Are those centers replicated on the right side and the left side of the brain?
In general, they're on both sides. That's right. There are some differences, but in general, they're pairs. In the case of the visual system, the left half of your brain processes information in the right side of the world, and the right part of your brain processes information in the left side of the world. And there's a place down the middle where they communicate, and they do it both sides. So in that particular case, it has to be on both sides.
Question back here. In your map, are there places in the brain for music and mathematics, for example?
There are parts of the brain that are necessary for music and mathematics. There are parts of the brain that are very specific in ways that you wouldn't believe. So, for example, down here, sort of in the bottom, there is a face area. And if you have a lesion in the face area on both sides, then you have a condition called prosopagnosia. What that means is that you can see faces. You can tell if the face is a happy face or a sad face. You can know where the nose is. If they're glasses, you say they're glasses. Or if the person is bald, you can say they're bald. However, you can't recognize faces. You can't even recognize your own face. You have no idea whose face it is. You just can't quite tell. And actually, the ability to recognize faces is, like everything else, has a very wide range for normal humans. Some people are wonderful at recognizing faces, and other people are not. I'm not so good at it, actually. And I used to teach medical students before I came to Salk, and I taught them in small groups of about 8, and every year there would be 2 people who I could never quite, I knew who they were, I could tell they were different, I knew their names, but I always got their faces confused. But many people, I'm on the bad end of things, but many people can remember faces incredibly well. And so, and this is true of almost all areas. For the people in this room, this is something I mentioned to somebody before, for the people in this room, this part of the brain here, visual cortex, is called V1, and there are 36 visual areas. This is another one over here. V1, if we took the people in this room and counted the number of nerve cells and the size of their V1, we would find that it varied about 3-fold. So some of you have a V1 that's 3 times bigger than the other ones. There's somebody in this room that has a V1 that's smaller than the V1 of an average chimpanzee. Okay, but whoever that is, it's not that bad, because the total number of neurons you have in your brain from individual to individual changes a little according to body size and so forth, but not that much. And if this changes a lot, that means if this gets small, that means something else has to get big to make up for it. And this is not known, but I'm betting that that if we looked at, I've had friends that I've tried to get them to do this experiment. We can image brain areas to see how big they are. You can identify these areas in imaging like you have for. If you bump your head like you have a brain scan. And I'm betting that baseball players are going to have big visual areas. There's an area up here it's called MT, called middle temporal. And we know what that does, and it is for detecting motion. And what a baseball player has to do is they have to hit a ball that's coming at them almost 100 miles an hour. They have a few fraction of a second to figure out what the trajectory is and project that trajectory to where it's going to go, and then they have to coordinate. And things like that are on the borderline of what people can do, what it's possible for humans to do. And I'm betting that those people have bigger visual cortex, bigger MTs, and things like that. I think they would have to. But there are many things that we do that not all of us, most of us not, but there are many things that humans do that are at the very borderline of what it's possible to do. Playing a Mozart piano concerto, it's incredible. And most people can't do it. Most people could never do it, actually, as well as the people who do it extremely well. And they probably have modifications in their brain
Question?
that permit this. Yes, please.
So are the sizes of these areas or the number of neurons in any given area an acquired trait, or is it something you're either born with if you'd be one or not?
Yeah. So it's not known. It's not known in all the cases, but it has been studied, and there are studies in twins. It might be genetic, but hard to detect. You see, that's why you don't know. But in twins, they've looked and see what parts of the brain are the same in identical twins, and the frontal part of the brain tends to be much more similar in people who are closely related and more separately related, more distantly related. Yes, please.
Just two quick questions, and that hopefully you can get on. Are there two language centers because one of them is your native language and other is for learned language? And second question, what is the difference in language between left and the right side of the language processing?
Okay, so the first thing is that there's just one language center, but different parts of language are distributed in different ways there, and it's hard to, and I don't know so much about it, and not so much is known. But what I do know is that if you learn a language late in life, it's overlapping in the same area up here, but it takes a bigger area. It's not so compact, and the second thing is that language is one of the functions that tends to be lateralized, and it's usually on the part of the brain opposite your handedness. So it would be on the left side of your brain if you're right-handed. I mean, every one of the things I've talked about is something that could be talked about for a long time, because there are lots of things that are known. There's studies of all these things. There are lots of things that are known about it. This is just a kind of a quick survey. Okay, so I'm going to stop right now, and I'm about to start telling you what's in here. So let me just make sure there are no more questions about this first part. Yes, please.
I have a quick question. The front part of the brain, some modern day theologians feel that they call that the angel brain. The part of the brain that is the least known about and not explored as much, and it has to do with metaphysics. What do you say about that? Have there been MRI studies or studies done to watch the formation of those thoughts?
Yes. So generally speaking, the fronts of your brain have to do with executive functions. All the parts back here have to do with movement and sensation and audition, skin senses, vision, and so forth. That stuff isn't in the front. There are things that are related to that in the front, but these things are back here. And if you look at a mouse brain, vision is a little exception, but most of the time you can't even tell. You can barely tell a mouse brain from us. In other words, all the rules are the same. We have bigger frontal lobes than other animals, and those have to do with executive functions, with making decisions, with making judgments about things like that. And there used to be, in fact, it was a Nobel Prize for it, for performing frontal lobotomies on people who were in psychiatric hospitals. And so we know what people are like if they don't have their frontal lobes, and their personalities are different. They make very bad judgments. A friend of mine who's a neurologist had to testify in a trial for this guy who got in all sorts of trouble because he ended up I don't know, marrying a prostitute or something like that. He lost all his money. He had broken some laws along the way. He wasn't a bad person, but he just made a lot of mistakes. And my friend, a guy named Tony Damasio, who's at USC, testified in his trial and was saying that he should not be held responsible for this because he he had this giant tumor that was wiping out his frontal lobes. He had been fine in the early part of his life, and then when this tumor had damaged his frontal lobes, he lost his judgment and his personality totally changed. So, yes, Sarah.
Are there overlaps or correlations between abilities, say, in music or mathematics or things like that in the brain that can be identified?
So mathematics and music and speech all involve receiving, and so this part of the language area is sort of the receptive part, and this part is sort of the speaking part, and if you have lesions here and there, you have language deficits, but they tend to be in two completely different categories. The ones here are ones where I would say to you, is this a piece of chalk? Or I'm going to show you a piece of chalk, and you would say, and you would not be able to understand the word chalk. That's called a receptive aphasia. If you have a lesion here, like for the stroke, you know it's a piece of chalk. You can write down the word chalk, but you can't come up with the word chalk and say it. And so they're spread out that way. But these language areas are, and all other language areas, have a lot to do with all kinds of symbolic manipulations. And so when you're doing mathematics, you're using probably the other side of your brain from the side that you normally use. Something David I know knows, and everybody who works with equations knows, that you have a problem you don't know how to do. You have to do some fancy integral, you don't know how to do it. You go to bed, you wake up the next morning, you know the answer. It's not that you know the answer, you know how to do it the next morning. And oftentimes, if it's a hard problem, you don't sleep so well, you're kind of restless that night. But you're not thinking about the problem specifically. I mean, maybe off and on, but you're not making any progress. And so somehow or other, your mind is symbolically grinding away there all night and presents you with the answer the next morning. And that's not normal language. That's going to be language. And I have a friend who thinks about things, and after she's thought a problem through, she changes it into language and explains it to you. And I can't quite understand this, but I understand that Einstein tended to be that way too. He tended to think symbolically. Yes?
I have a slight, you keep on referring to personal pronoun, you, me, I. And I'm sure you don't really mean that.
Well, sometimes I do.
No, you know what I mean.
People in general, yeah.
What?
I sometimes personalize the people in general.
No, I mean the center of consciousness.
Oh.
When you refer to you, me,
yeah.
You're always referring to the conscious
yes.
mind.
Yes.
Whatever that is.
Yes. Yes.
So I'm just a little bothered by you know, assuming something I thought you wouldn't assume, that there was this you there.
I'm probably not assuming anything you would object to, David, about that. I'm speaking colloquially.
This business of who it is that's thinking during the night.
Yes.
It's not you, but it's the mind.
Yes, yes. Well, or it's the person who's solving the problem during the brain that's solving the problem during the night. Is what I would say. I wasn't meaning anything deep about it. I was just speaking colloquially. Yes, please. Yeah, up there, and then I'll cut to you.
I read a book called How We Decide, and he makes a strong case for the importance of emotion in making good decisions in areas that were traditionally thought of as logic being more important, going through steps 1 through 100. And I'm just wondering, is that because of different areas of the brain being affected, or?
That's the sort of thing that people don't know about. It's definitely true. In science, people, what's more logical than science, right? However, anybody who does science does it basically because they have an intuition that they want to work in this area, they want to work on this problem. They don't know what the answer is, but they just have this feeling that this is going to be good. And some people are lucky over and over and over again, and some people are never lucky. Yes, please.
The other thing you mentioned is that by doing brain imaging and seeing what areas of the brain light up, they could tell what decisions people are going to make before they make them.
Yes.
That's
that's it. So were I to have given a different talk tonight, Sarah wanted me to tell her a talk title in advance, and so I did, and I thought I better stick with it. But what I probably would have done is talk to you about the techniques that are being discussed in the workshop that I'm at, because it's absolutely amazing. The things that a lot of things we can't do. We're a long ways away. I think we still have job security, but it's just amazing what can be done these days. Yes, and you had a question.
Early programming in computers, you used to work in a thing called machine language, which was dealing with the ones, zeros and ones. Whereas computers have developed, you're going in more into higher level languages.
Sure.
Does the brain happen to work in a brain level language as compared to a higher level of awareness, I guess?
So I'm actually going to talk about that, because that's one of the things that's very different between computers and brains. And so I'll get to that in a second. Sarah, I didn't notice what time I started.
About 6:30.
6:30. So I'm supposed to finish at about 7:15.
7:30.
7:30. Okay. Okay, so now I need to tell you about what's in here. And what's in here are nerve cells, and I'm going to draw some here. And every nerve cell has a cell body, and in the cell body there is the DNA that we all have in all of our cells. And sticking up from the cell are these long, and they look like lines here, but they actually are tubes, little hollow tubes that are very thin, about a tenth of about a hundredth of the width of a human hair, and this is the cell body of a cell. Every cell in the body has a cell body. These are dendrites, called dendrites. These and those. And then coming out down here, there is a long tube, again, less than a hundredth of a thickness of a hair, and this is called the axon. So you have to know from what there's going to be 4 words you have to know: cell body, dendrites, axons, because every cell has a cell body. When I count neurons, I count the number of cell bodies. But then they all have these dendrites sticking up and the axon going out. So this is cell 1, and here comes cell 2. These are the dendrites down here. The more dendrites up here. DNA's in there, another axon going someplace. Okay, this is cell 2. And the way neural circuits are formed is that information comes, in a way I'm going to say in a second, comes into the dendrites, it gets added up, gets turned into pulses, nerve impulses. Those nerve impulses travel out this axon, and they go over here, and they touch the axon here of the white cell, number 1, touches the dendrite of cell number 2, and the place that it touches is called a synapse. The synapses are very fancy, and I'm going to tell you a little bit about them. I spent a good fraction of my career working on synapses, but I no longer work on them now. But they're interesting for a lot of ways. One of the reasons they're interesting is because every single drug that affects the mind works by changing, in one way or other, the way synapses function, with no exceptions. all the drugs of abuse, alcohol, marijuana, Valium, Ambien, they all work on synapses of one sort or other. Many sorts of synapses I'm not going to talk about. Now, there is something terribly wrong with this, and this is the way a neural circuit is made. Cell number 1 gets information over its dendrites, it changes it into nerve impulses. Those nerve impulses go over to cell number 2, and the information in them is communicated to cell number 2 at the synapse. Forward, cell body, axon, dendrite, synapses. Now, what I've just drawn here is horribly misleading for the following reason. Cell 1 and cell 2 both get and receive about 10,000 synapses. If you look along this axon, very, very close together here, about a tenth or so of a mil, a thousandth of an inch. There's a synapse all along here, and this axon branches a lot, spreads information over a big region, and in fact, something I didn't tell you about these areas are every area has a map. The visual system has a map of the world on it. So if you're looking at the world, there's a place in the brain here, if you're looking at something over there, the nerve impulses from that place go to a place here. The nerve impulses from the part of the world just next to it go just next to it up there. And so there's a map of the world up here, and these axons branch out and send information to about 10,000 cells spread over a particular part of the map. This cell here is getting information from 10,000. On average, about once a second, maybe 10 per second, the information is coming over all 10,000 synapses. That information goes along here, gets spread out, and sampled by the dendrites of 10,000 other cells. So these neural circuits are made by complex networks like this, where there are 10,000 of these and 10,000 of those, and really many more, and they're sending the information over this map. Okay, so already we can say something about the comparison between brains and computers, and I'm going to do that over here. And up here are the brains, and over here is the computer. And the brain, and in the brain there are about 10 billion nerve cells in your cortex. That's 10 billion of these fellows. And in the brain there are, I have to translate a little bit here, there are 100 trillion synapses. Let me just think for a second. 10 to the 14 is a billion. I think that's right. Yeah. Okay. 10 trillion synapses, or 10 to the 14th. 100 trillion. What was it? Thank you. 100 trillion synapses.
10 to the 14th. It's easier.
Yeah. Right. and the wires that are hooking them together, these axons and these dendrites, you added them all up, their total length is between 200 and 250,000 miles. If you took all the wire in your brain and you strung it out, it would go from here to the moon, on average. Now, a computer doesn't have nerve cells. It has circuits, and they're kind of variable, and it's hard to describe how big they are. But the thing that's closest in the computer to 100 trillion, right? The thing that's closest in the computer to synapses are transistors, and a modern computer chip has 1 billion transistors and 18 miles of wire. Now, if I had written that down first, you would have been impressed. I'm really impressed by that, because that's not very big. It's like that big. It's very thin. They get 1 billion transistors on there now, and the width of the wires in the computer chip and the width of the wires in your brain are not that different. They're about the same these days. However, if I'd written that down first, then you really would have been impressed with this, because this is one of the main differences between computers and brains, and that is that we have just so many more computing elements than a computer does, and that's part of our power. Yes.
But the processing strategy between those two, the computer and the brain, is night and day.
Totally different. Right.
One is linear programming. It may branch,
Yep.
but it's linear basically. And the other one, it appears, is something else.
Yes, I'm going to talk about
Very
that.
good.
Yeah, I'm going to talk about that. Now, if I had talked about a different thing, like how fast the computer does it and how fast the brain does it, the computers are vastly faster. People talk about gigahertz clocks in computers. We don't actually have a clock, but it would be nothing like. But it would be nothing like as fast as that. Our time scale is in kilohertz, which is 1,000 cycles per second, rather than 1 billion cycles per second. So it's very different. But there are other ways that are different that I'm going to be saying. Okay, the next thing is I need to tell you something about these synapses. I'm going to tell you two things. I already told you one, which is interesting, but I'm not telling you much about it. That is, all the drugs work there. The two things are that your memories are stored by changes in these synapses. So when you store a memory by things that are partly understood and mostly not understood, I mean, we know lots of little bits, but not everything. When you store a memory in a synapse, that memory is stored in some changes in the synapse, and I'll tell you a little bit about that in a second. But the changes are basically how effective the synapse is, how well it transmits information from cell 1 to cell 2, and so all of your memories are stored, spread out all over the brain in millions of synapses. Okay, so what's different then between brain and computer is that memory is embedded in the brain. What I mean by that is that instead of and it's separate in computers. You have a CPU that does all the calculations, and it has a little bit of memory there, a cache memory, so it can get its stuff fast. But then over here, they have all the memory chips. And what the computer has to do is it has to go over, get an instruction, bring it back, and execute it. And what we do is we have our memories embedded right in the circuits that are doing the computation. And so this is what you were saying. You don't have a program. You don't have a program like that. It's not as if you have a program someplace and it runs. It doesn't work that way. What we have is we have our programs embedded right in the circuits that are doing the calculations. Okay, so one thing that's interesting about synapses is memory. The other thing that's interesting about synapses is that they are unreliable. And they're made that way on purpose. They have to be that way. They work. They're not unreliable on average, but at any time a nerve impulse comes, they either do or don't send the information, but it's totally probabilistic. It's totally random. You can't predict. It's like flipping coins. If you flip a coin, a fair coin, you never know if it's going to come up heads or tails. But if you flip it 1,000 times, you know you're going to get very close to 500 heads and 500 tails. And that's the way these synapses work. The sort of average for synapses is that if you have a nerve impulse in, if you have 5 nerve impulses in, it transmits the information about those from cell 2 about 1 time. In other words, about 20% of the time when a nerve impulse comes, the neuron number 2 doesn't hear about it. 1 time out of 5, neuron number 2 does hear about it. And so in terms of I have a defect, and I have many defects. I've mentioned some tonight. One of them is if I'm close to the board, I can't read the words. Okay, the reliability. The brain is very unreliable, and computers are very, very reliable. That's not to say they don't break sometimes, but the number of operations that you do with the computer between failures is incredibly low. It's just hard to believe you could even manage to do that. There's a reason why you make these unreliable synapses, and that is something that the brain does that the computers don't do in a very small way. When they send astronauts up into space, they send 3 computers with them, all doing the same thing. So if one breaks, they still have 2 left. We do the same thing on a big scale. We have a lot of redundant information. So cell 1 sends cell 2 information, but there are 100 or 1,000 other cells like cell 1 that are doing the same thing and sending the same information to cell 2. And what this means is that if one of those 100 cells over here dies, it doesn't do anything to the brain. If you kill cells at random, then nothing happens. It has no effect at all on the circuitry. And in fact, if you kill cells at random, you can kill a lot of them and still have very high levels of functioning. And we were talking before about President Reagan, and my speculation is that he probably had lost, by the time he finished being the president, he'd probably lost a quarter of his neurons, and yet he still functioned at a very high level. So, and any of us can lose lots of synapses, lots of neurons, and our brain works just fine. I shouldn't be saying this, but we all, starting at age, 2 things. You're probably worried about your nerve cells dying. So I just told you, it's okay if a few of them die. The fact is that for healthy people, almost no nerve cells die. If you take an 80-year-old person or a 90-year-old person and they're healthy and you count the nerve cells and you count them in a 20-year-old person, same numbers, it's no different. That's the good news. The bad news is that about 0.5% of your synapses go away a year. That's not true for everybody. I mean, it's true in general, on average, but individuals, it happens at different rates. And so part of the reason that our memories become less good as we get older are because the hard disk is getting full. It takes time to find it and find the memories and so forth. You have more memories, but part of it is that you're losing synapses. Yeah.
I thought there was evidence that you actually can grow new synapses.
Yes, you can grow new synapses, and synapses are constantly at a low rate turning over. But if you just look at the total number, if you do brain scans, what happens is that the cortex gets thinner over age. It's very systematic. It gets thinner over age. The number of cells aren't changing. The reason it's getting thinner is the synapses are going away, and some of the wire hooking them up is going away. Yes?
Why are the dendrites and axons tubes?
Oh, it's because the whole cell is covered with a surface membrane to keep the outside world different from the inside. Inside the cell, different from the outside the cell. So it's extremely well known. It's like a grape. So there's an inside to the grape and the outside of the grape. The grape is covered with a skin, and all cells in your body are covered by a skin made of a phospholipid membrane, and it keeps the stuff on the inside from getting away, and doesn't let stuff on the outside that shouldn't be there come in. But if you're going to have a fancy shaped cell like this, then you have to have it covered with membrane every place, and have the outside and the inside separated. That's why.
Question.
Yes.
What is meant by the phrase, we only use a tiny, tiny fraction of our brain power?
Yes. There are certain things that are standard truisms from folk neurobiology. One of them is we only use 10% of our brain. Another one is every time you take a drink, 100 nerve cells die, and neither one of them are true. And I'll come back in a second. We're using all of our brain all the time. We have a lot of spare capacity because we can it's quite flexible up there, but so we can lose part and then take over part of that capacity, but we're using it all the time. Yes.
Other than an infant is born with many more neurons or synapses in the early years, vast numbers pruned away?
Yes. So when babies are born, they have all the nerve cells they're ever going to have, with a couple of tiny exceptions, but they have only maybe 20% of the synapses they're going to have. The reason a baby is a vegetable for the first 3 months is because it doesn't have any circuits in his cortex yet. When it starts smiling, that's when it's getting some neural circuits up there, and they're starting to work. Babies, when they're the first 3 months, are making 1 billion new synapses a day. They're making enormous numbers. The circuits are growing. It peaks at sort of adolescent age or something like that, and then it goes up like that. There may be there's rearrangements, but then it starts this, when you're 18 or 15 or something, it starts this slow decline that goes on average the rest of your life. Yeah. Okay, so. The reliability is very low, but the fault tolerance is very high, and it's very low for computers. You can often have a sticky bit someplace and it doesn't bother you, but a very small change in a computer functioning can wipe everything out, and that's just not true for the brain. We're different in this, this is just a very brief aside, we're different from this than insects. Insects have one nerve cell for a thing, and what they do is the insects make a lot of copies, and they hope the copy from one DNA gets through. we make very few copies, but we make the best possible versions of the copies we make, so the DNA is likely to get through. And one of the ways we do that is by having a lot of redundancy, which gives us a lot of fault tolerance. Okay, the next thing is that there are a few computers that process things in parallel, where most computers do things in sequence. They get a piece of information, they do something with it, they get another piece of information, they compare them, they go out and get another one, they keep doing it like that. There's a clock that says do this, do this, do this. There are a few computers that have a job where they can break the job down into 256 parallel jobs and do them all. So a company called NVIDIA makes image processors that uses 256 or 512 little tiny computers, and they process images for computers with all of those, and doing the same thing for all of them at the same time, taking different pieces of information. We do that massively. Our cortex, our visual cortex, has a whole image in there. That whole image, gotten from the world, represented in nerve activity, that whole image is processed the same time. And so even though our brain is very slow for individual things, it's massively parallel, and so because of that massive parallelism, we do things very quickly.
So given that a computer chip is a tiny fraction of the mass of the cortex, if you were able to connect multiple chips to more or less be equivalent to the mass of a human cortex, and connect them in a way to make it possible to process parallel information, could you mimic that process with a computer?
No. And so, for example, your computer at home has more computing power than a fly, but your computer at home can't process images the way a fly does, and you can't make it do it. And I'm going to come back to that in just one second. Parallelism. Okay, our computers are very parallel. Our brains are very parallel, and there's low parallelism. There is some, all the new things you can buy now. Most of the chips that are being sold now have 2 computers on them, or 4 computers. And inside the computer, what they do is they save time by looking ahead and getting information they're going to need. And so at the same time they're getting a piece of information they're going to need in the future, they're doing something with what they already have. So they're doing things in parallel. So that's one of the ways they've been being speeded up. But it doesn't come close to what we do. Okay, the last thing is, in order for evolution to work, we have to have structures that are scalable. What that means is, our hands are not like a mouse's hands or a squirrel's hands, but they're pretty similar. They're very homologous. A squirrel can pick up things with their hands. They don't have the fine control we have, but they can pick things up. A baby has tiny hands, and a really large person has very big hands, and they all have to work perfectly as the animal gets bigger, but because they have the same job to do, and so you have to design them so it doesn't matter how big you make them, they still work. That's not so hard with hands, but it's very hard with computer circuits. And our circuits have to have a scalable architecture. What that means is, for evolution to work, what that means is, a scalable architecture means that you can make the computer more powerful just by making it bigger. You don't have to redesign anything. Every time Intel changes anything in a chip, it's a giant job to redesign the whole thing. They have to, even if they only make the wires thinner, they have to redesign the whole thing. We the reason the person in here who has the tiny V1 can still see is just about the same as the person with the big V1 is the person with the tiny V1 has the same processor. Everything is doing the same. It just doesn't. That person just doesn't handle quite as much information, quite as much resolution as the person with the big processor, but it's the same processor, just bigger and smaller. And so the last difference I'm going to talk about is scalability. And our brain is very, very scalable, and computers are not. Not that they're not scalable at all, but in general, there are no good computers that are scalable. People are trying hard to do that. People have tried to do it in the past. Trouble is that we don't know how to program efficiently parallel computers. We don't know how to make them. We know how to make things that are parallel, but we don't know how to do it. Okay, so I'm going to answer. I'll stay in as long as you guys want and answer questions. I'm going to let everyone go in 5 minutes, but I'm just going to say very briefly, what does physics have to do with this? So let me tell you what I'm working on, what I've been working on for the last few years, and that is I'm trying to answer the question, what are the design principles that brains use that give them a scalable architecture? There's got to be things about brain circuits that let them be so easily scalable. And this will almost certainly not be of any practical value in my lifetime. But if it were of practical value, it wouldn't be because I was curing Alzheimer's disease. It would be because I would be able to tell Intel or ARM or somebody, NVIDIA, how to make better computers by making use of principles that the brain uses, so that they could make computers that were more scalable, and that way they could make them more powerful just by being bigger. And somebody over here asked the question about why your computer couldn't do it so well, and that is that we can't really be able, until we understand better what the brain does, there's no way that we're going to be able to program a computer to do it as well. We're quite clever at figuring things out, special problems. We can solve those wonderfully, but in a general way, we can't do it without understanding what the principles are. So one of the things that needs to happen before we can do better computing, that's not why I do this, by the way, but it would be a practical thing if it worked. One of the things we could do better if we had is that we could design better computers by making use of principles the brain uses. Where does physics come in? Well, for understanding a scalable architecture, then you can't just say, oh, I'm going to study a scalable architecture and understand it. You can't do that. What you have to do is you have to use theory. You have to have a lot of facts. It's not theory that doesn't relate to anything. It's theory that relates to specific observations. But scalability is something that physics is really good at studying. And there are a lot of scalable things that are interesting in physics. And absolutely the most beautiful and most effective theory in the world comes from physics. So if a biologist wants to do theory, then what they have to do is, if they're going to do it well, is they have to know as much theory as a physicist does, ideally, so they aren't going to be limited by their tools. They'll have a wide range of things to do. And I could give you a specific talk about this. I could give you a specific example of how you use theoretical approaches which are not making computer models, they're not making mathematical models, they're a physics-style theory. I could give you a specific example of how you do that to discover design principles that brains use to make a scalable architecture. But it would be too much for one evening anyway, and it would take a bit of time. Okay, so I'm going to answer, but maybe we should give. There are going to be people who have things they have to do, and they may not want to stay, but they may have things they they need to go to. So anybody who needs to should go now, because it's 7:30. Hang around and talk to anybody who still has questions.
I actually was hoping you'd explain the scalability just a little bit, because I understand the notion of a hand, but it really does the same thing when the bones and the muscles are smaller as when it's larger.
Yes, that's right.
The brain has the same number of cells all the time. Is this scalability the addition of synapses? Because the brain is not physically.
Okay, so our brain has the same number of cells when we're born as when we die, pretty much. I mean, it doesn't have to, but in general it does. So the scalability comes because there's variability in the brain, and some people have a big area for one thing, and other people have a big area for something else. And if you're going to have different sized areas, you've got to be able to make the computer get bigger and smaller. But the way you study it is, the way I study it is, one way I study it is by studying fish. The reason for studying fish is that there are two ways, basically. One way is to study evolution. And you can look at a chimpanzee brain and our brain, and certain parts do exactly the same thing. You can study them. It's okay to compare them. The other way to do it is to study fish. And fish are not us, and they don't do the same things, but we're related to them, and a lot of the things they do are just what we do, their brains. And so with a fish never stops growing. It's born and it grows its entire life, and unlike us, it gets bigger, of course, and every year it grows. It never stops growing, and a big fish is an old fish. But the whole fish grows, not just the body. The brain grows, the eye grows, and it uses the same circuits, but the brain makes new nerve cells, unlike us, and gets bigger. And so by studying fish, you have an example where you can try to find a design principle that was used, and then the job is to figure out, how do I take this design principle and test it to see if it works for brains that we care about. Yeah. Yes?
Does gender affect the composition of brain?
Careful.
I have no personal opinion on this. No, it actually does, and it does in very interesting ways you wouldn't expect. I have a good friend who is a professor. I'm going to give you one example. A good friend who is a professor at Stanford. He was born genetically a woman. He was a woman when I first knew him, but he never felt like a woman. He didn't like to play with dolls. He didn't feel like a woman at all. He felt like he was a man trapped in a woman's body. And so after he became a professor at Stanford, he changed sexes. He didn't have a sex change. It's more complicated. When he was a she, he had breast cancer, which he inherited a BRCA1 gene, which is a bad gene from his mother, had cancer in both breasts, had them removed. And I remember I saw he was she then. Right after that, I said, "Gee, that must be very disturbing." He said, "No, glad to be rid of him." Anyway, so he started. I got email from him, and he said that he was going to a conference, but he was changing from a female to a male, and he was starting to take testosterone injections, and he was worried everyone was going to reject him. And nobody did. Everybody was totally happy with it. One of my friends who was the chairman of this conference he was going to wrote back to him. I wrote saying, "Oh, it doesn't matter," something like that. She wrote back to him, "This is terrible. I have gone to great lengths to get the gender balance for this conference just right, and now you've screwed it up. It was really good. Anyway, so what he told me was that they did some studies on him before and after he some psychological studies before and after he started taking testosterone. His beard started growing, and he started wearing men's clothes. He sent me pictures of himself with his first tie, and I also had and also a picture when he was in a sort of a fancy dress for a wedding, and it was his first tie. Anyway, he said that they told him two things were going to happen. One was that his sense of where he was in space was going to improve, and his ability for spatial manipulations with his brain, and that he would cry more. And he did this. He used to lose his car in the parking lot. He never loses it now, and he never asks for directions. That may be different. And he also says he also cries. He cries now. You know, go to a sad movie, and where he wouldn't cry before, now he does. So these are brain things. So there are differences, and they come just from hormones. It's not genes in that case. Yes.
You just answered. You're saying all the brain differences between men and women are because of different hormones.
No, there are probably some genetic differences too. So there is a phenomenon called imprinting, which is there's two meanings for that, but one of them is that there are genes that the mother and the father imprint, and then you may get a mother's gene and a father's gene, but the mother's gene will get used. What they did is they made a mouse that didn't do that. They have a tricky way of doing it. They made a mouse that didn't do that, so they used only father's genes or only mother's genes. What they got was, when they used only father's genes, they got a small brain and a big body. And when they used only mother's genes, they got a small body and a big brain. Anyway, so there are some differences like that are not that are genetic, but they're not sort of Mendelian genetic.
He's had a question for a while.
Oh, I'm sorry. Yes. Oh, I'm sorry. I wasn't looking over here. Let me stand over here.
Most cells in the body replace themselves. Do brain cells replace themselves?
Yes, some do, but almost all not. This is something there's been a dispute about, and there are several replaces. Okay, so if you have an axon, the cells are all in your brain. Your muscles are down here. What they do is they send a big long axon down and make the muscles contract here. If you cut that nerve, and you don't cut it, but you don't disrupt it, you have it touching each other still, it'll grow back. So in that sense, they regenerate. If you kill cells in your olfactory system, or in your taste system, in the periphery, in your nose, those grow back. And there's a particular part of the brain where cells are made your entire life. That's a part of the brain called the hippocampus, and there's one part of that where there's cells that are being generated. It's almost certain that there are no other cells that are being made. All of your cortex, you have what you're born with, and but that's not true for fish or salamanders. They can grow cells back.
So these synapses that are formed are fairly permanent. They don't change.
They can change. They change all your life. In certain ways. So if you have a brain lesion, the synapses aren't just gone there. The cells that are sending synapses will send them to neighboring places, and so the synapses will grow back to a certain extent. And also throughout your life, they get their size gets changed. They get bigger and smaller, and that makes them stronger and weaker. And that's part of the way memories are stored. They're stored in that. So those things change. There's a lot of plasticity up there, but there's not. You can't just grow it back.
Is there any? Since you're talking about changes over life, is there any? I mean, the tasks and memory are stored in the synapses, which are changing.
Yeah.
Is there any way of knowing about how that plasticity changes over time? The ability to change synapses.
Yes. There is some data, this is not something I'm an expert in, but there's some data in rats that things that are related to this process. There are laboratory ways of studying changes in synapses, and it has been claimed that those there's less plasticity in old mice or rats than young ones.
Didn't Marion Diamond sort of study that at Berkeley? She used to always, we used to call her the brain lady when I was growing up. She always used to say that the most important way to preserve your ability to make synapses was to continue to use your brain, and that people who had very little stimulation and were not actively doing puzzles, whatever, were the people who had the most difficulty with that kind of plasticity. And I wonder if that's really true.
Yeah, it's hard to study, but it's definitely true that people who are mentally active and are interested in things like that do a lot better with age.
And even mice, you could.
And with mice too, that's true. That's true. So if you have a mouse or a rat and you raise one of them in an impoverished environment, just a box with 4 sides and water and food, and another one with other mice or rats, toys, things to climb on, things to burrow in, the ones that grow up in the enriched environment will make many more synapses than the ones that are in the impoverished environment. That's true. Yes.
I had read that there was a change, an increase in the mass of the cortex in the early teen years, like 11 to 15, where is that adding cells or what?
No, that would be adding synapses.
Oh, okay.
So your cells are there, unless you have a tumor. Your cells are there, but then changes in the size of the brain are all changes in the circuits. The number of synapses, the number of wires, those go down and up. And so as you age, as one ages, and your cortex gets thinner, that's synapses going away. Yes.
From an evolutionary perspective, why did the brain develop into 2 halves rather than sort of a monolithic structure? And why are the halves reversed relative to the sides of the body?
So the reason it develops, I don't know the answer, but I can tell you some things about it. In general, there are a few exceptions, but we're bilaterally symmetric, and we have been forever. And so when you design a new species, you don't start from scratch. You have to start with what you had before and modify it. And there are genes that are called transcription factors or homeobox genes and things like that. And what these genes do is they regulate a whole program of a whole lot of other genes. And when you change with evolution, when you change something, you just tinker with those. And so basically the design is the same. So I think that we had to have an eye on each side so we can see, originally they were on the sides of your head so you could look in both directions for danger, and so you had to have processes for them, and so they just double the eyes. They just made one on each side and kept the bilateral symmetry. Okay. Why do we do things that, why do we have designs of the brain that we think are dumb? For example, your eyes are in the front of your head, and the visual system starts in the back of your head. Why would you not? Why don't you turn it around and put the visual system in the front of the brain? The reason is that the eyes are going. The information has to go all the way to the back and then go back to the front to do everything. The reason is that when our ancestors, when the fish or early bilateral animals developed, the eyes were on the sides of their head, right, and the brain was right underneath there, and so the eyes were as close to the part of the brain that did vision as possible. Then, when it became evolutionarily possible to move the eyes around front and you have better vision, like us or primates or cats, you can't just redesign the whole brain. You're stuck with what you have. And so what they did is they moved it in front, and the gains, apparently, I mean, this is circular, but the idea is that the gains that you got from having the eyes in front beat out the penalties of having to go back to the back of the brain.
And what about the reversal on the left versus right?
Okay, so one of the things that you want to do if you're say a fish is if you want to get away from something on this side, you want to flip the tail the opposite way to push you away from it. And so some things are crossed and some things aren't, but you originally was designed apparently to have it so that your escape response was on the same side. That you crossed over to make the escape response as fast as possible. So you didn't have to you had to cross someplace to get it to the other side. You could have sent to the other side of the brain and told it flip your tail, but it was faster to have it cross right away. So it's something like that. It's something we're stuck with from the way we originally developed. Yes, Sarah.
Is there a difference in the way the brain functions during sleep, in terms of its design structure, and how do we measure consciousness versus when you're dreaming? Are you conscious in a different structure?
Yeah. Gee, so sleep is something. There are certain things that I should know a lot more about than I do, and I know something about sleep, but I'm nothing like an expert in it. But there are, I'll tell you, I can in about 4 sentences tell you everything I know. One thing is there are different levels of sleep, there are different states of sleep, and your dreams come in a part of sleep that is called REM sleep, for rapid eye movement sleep, and nobody knows why you sleep. They don't know what happened. They know it's not good for you if you don't sleep. But no one knows why it's necessary to sleep. All organisms, as far as I know, all multicellular organisms, I think all the ones that have any behavior sleep. Flies sleep. Some animals sleep on one side of the brain while the other side has to keep awake because they live in a dangerous environment out in the ocean or something like that. And there's a lot of evidence these days that one of the functions of sleep, it can't be the only one, but one of the functions of sleep is that memories are consolidated during sleep. So if you there's some really fascinating studies. This would be another lecture, But when a rat runs through a maze and you record from cells in a certain part of the brain, when the animal is at certain points in that cell, in that maze, certain cells will fire. And so the firing pattern, as the animal moves through the maze, moves around in the brain. If you record from those same cells when the animal's asleep, he will play back the same firing patterns, the same patterns that he was doing as he was running through. And if you disrupt that, then they don't remember as well. And there are a number of studies that show if you keep people from, you make them sleepy, but let them give them some sleep, but have them be equally sleepy somehow, that they remember better. Yes.
I have a question, but he's been waiting
sorry.
for a long time.
I keep wandering over this way, and I'm neglecting the guys over here. Yeah.
Thanks. So you and Professor Groths both mentioned this KITP program related to frontiers and brain imaging. So I want to ask about a specific brain imaging fantasy I have. So I have this fantasy that I can get a sensor hat and that I can compose music in my head, and this little sensor will read it out and go into a computer or MIDI. And one could also imagine lots of applications where you picture images in your head and they go into computers. So I wanted to ask, what are the prospects of something like that, and what are the barriers that we have now to achieving something like that?
Okay, so what can you do now? Right now, you can have, we're going to have a talk tomorrow that has a rat running around in the environment and looking at things. And while he's doing that, there's a camera on his head that is looking at his eyes to see where he's looking. There is a microscope that is taking images of all of the cells in a region of his visual cortex, and they have a dye in them that lights up when the cells are active, so you can follow the activity of every cell. You know exactly where the animal is in the environment. He has a little hat with lights on it, so they can follow those. You have pictures of his eyes, and so when this data are processed, then you can see over here what the cells are doing, over here what all the cells are doing. Over here you can see a picture of what the rat is doing. Over here you can see a picture of what the rat is seeing. Over here is a picture of the world with the rat running around in it, with the part of his brain that is looking at a particular location being projected as a red circle up on the world. So that's the current state of things. Now, I don't think you ought to wait. I think maybe you ought to start learning to get better at writing it down or something, because it's going to be a long time before the rats that do this, they don't get out of there. They don't live through it, because you have to cut a hole in the head to get this resolution and everything, and so it's going to be a long time before we're to the point where things like that are what you say are possible. Yeah.
But something like artificial vision isn't so far.
But that's right. But something like artificial vision is already there are people who are paraplegic and they can't move, but they can record from their head, and they can move a mouse pointer around on the screen by thinking about it. So, and this is early days still. It's crude, but those things are coming. But it's much easier to do something specific like that than it is to take something really complex like something you're thinking about, a bunch of notes you're thinking about, and translating them into something we can get out. That's going to be a while. But and also you'd have to get a lot of information, and you can't get that information from 100 electrodes or something. It's too much information. Yes?
I think it's fascinating that the cells that do replicate, the neural cells that do replicate, are some of the most primitive parts of the brain, and that, in fact, it suggests that evolutionarily there's some good reason for you to not grow new brain cells.
Yes, that's
so
right.
What's the sort of evolutionary advantage of having cells that never replicate?
Okay, so I don't know, but I can tell you a speculation about it. So the cells that do replicate in us are cells in a particular part of the brain that lives a hard life, and cells there die at a high rate. You can't just change the number of cells and have it work. The number of cells in one circuit has to match the number of cells in the next circuit, has to match the number of cells in the next circuit. And so, this part of the brain where the new cells are made is one that's involved in forming memories and knowing about spatial locations and things. And so, if they died off so quickly, you wouldn't be able to function, because the whole part of the brain wouldn't be able to function. So, it's important that we replace those cells that are dying. Most of the cells don't die very fast, but these particular ones have characteristics where they do. Okay, but why not just have our cells grow back like a goldfish? And probably it's because we have a long life, and there are two reasons I can think of. One is that we have a long life, and and you don't want to have tumors growing up there. And over time, as time goes on, the chances of a change, enough changes accumulating to make a tumor increase, and so we preserve it that way. That's one idea. The second idea is that the brain uses things for other purposes than they were originally intended, and so it may be that if you made new cells all the time, or let cells dedifferentiate and divide and then come back again, that you would be taking over some of the functions that the brain is using for something else and it can't afford to give up. I've never looked seriously into this, but those are just two things I have, sort of random things I've thought about. Yes, please.
With the 5 nerve impulses in and 1 out kind of thing, is that an advantageous thing? Is that part of the programming? Or somebody who had, 5 nerve impulses in and 2 out, would that be an advantage?
So there are two reasons for it. The first reason is that in order to get fault tolerance, you have to have lots of inputs into a brain. Into every individual nerve cell, because you have to have redundancy to get fault tolerance. If you're going to have a lot of synapses on a brain, I'm a little tired. If you have a lot of synapses on a cell, in order to have enough to have redundancy, they have to be small because they won't fit on otherwise. If you make them small, then because of the way the machinery works, you can't make them reliable. They have to be unreliable if they're going to be small. That's one reason. The second reason is that you have to be able, you do it all the time, change the strength of a synapse by changing the probability that it's going to release, it's going to send information to the next cell. And these synapses are very fancy structures, and they remember back their past history of use, and they adjust their current probability that they're going to communicate according to that past history. And the way that you change the strength of a connection is by changing the probability that it's going to communicate. Because if you average over 100, then it doesn't matter if they're unreliable. All you do is look at it as the average, and that's quite reliable. So it's a way of very quickly changing how strong a synapse is. There's not another way you could do it that quickly because everything else is too slow. All you have limitations in the cell biological things that are possible. So that's one reason you want to do it that way. But the other reason is you have to do it that way in order to get the information in.
So I think we maybe should take 1 or 2 more questions.
Yeah.
Or try something is more silly. He's being very gracious. So I think he'll stay and talk to us privately, but I think we should let go at some point.
I have a question about neurological diseases that begin to eat away at certain parts of the brain. For example, with Alzheimer's or dementia, does it actually destroy the synapse, or is the synapse there and the destruction is in the sheath that covers the axon? And
okay, so it's both. I mean, it depends on different diseases. So multiple sclerosis has to do with the sheath being destroyed, and so the nerve impulses can't get from point A to point B because of that. And the reason it does that is it's believed to be an autoimmune disease where the immune cells attack the cells that are covering up the axons and make them not work anymore.
But the synapses are still intact.
The synapses would be intact in that case.
Okay.
In Alzheimer's, they don't know quite what we used to think we knew what caused Alzheimer's, but we're getting less and less sure these days, because it used to be that the theory that with Alzheimer's disease there's this protein that accumulates outside the cell and makes plaques out there, and it was thought that those plaques were toxic in killing cells. one of the drug companies just found out in a big trial that the best drug for keeping those plaques from forming didn't work. It made the people worse instead of better. And so it was, that's the way the drug company business is. You never know for sure. And so what people are thinking these days is that actually the real change in Alzheimer's is not the plaques. That they don't really matter. They're just a symptom of a sign of what's there. But that it's that there's certain things that are toxic that are killing synapses. And some of these same things that are related are killing the synapses.
So the synapse actually remains intact in an Alzheimer's patient?
No, they go away. They die. The synapses die. Lots of them die. Yeah.
When you said that drug and alcohol are attached to the synapse, does it destroy the synapse? And you said that's where the memory is. If the synapse is destroyed, does memory go away?
That's an experiment we can't do because the synapses are all over the place and we don't have a mechanism for killing all the synapses that are associated with one memory. That would be fantastic. It's an experimental tool. But alcohol actually makes just changes the way the synapses work transiently. And it doesn't, and it affects one particular class of synapses, and it doesn't change them permanently.
Except for with alcoholic dementia, right?
Oh yeah, but dementia probably isn't from the alcohol; it's probably from the diet. They probably just they drink everything. They don't.
So I think we should probably thank Chuck. He'd be next.